Got it, I guess. Okay. Well, let's get started. Doesn't want me to advance my slides for some reason. Oh, okay. So the outline for today's talk, I just want to sort of impress everybody about the insects as a group, just real briefly, and then go over just a couple of publications that provided some evidence of declines, and then talk a little bit about the difficulty of detecting declines, and then go into answering the question, are Maine insects in decline? And then if so, what might be some of the potential causes? Okay, well, insects, apparently there are over 1 million species that have been identified, 2 to 5 million, depending on whoever you're listening to, has that value as the estimated total of species that either exist now or used to exist in the world. 5,000 to 16,000 species described a year. So just a massive taxa. And I think this little pie chart sort of says it all. In the blue are the insects, so they make up half of the species richness on the planet. And followed by fungi, bacteria. Now, the microbiologists will say it's just because we haven't been able to identify all the bacteria and there's many more than insects, and that may be true. But as far as the identified living flora and fauna, the insects are the dominant group on this planet. And so we want to keep that in mind when we talk about the roles that insects play, both in ecosystem services, but also in promoting, human well-being. And I just popped down several of them out of my head, and I'm sure there are many more that people can think of, but wild plant pollination, crop production via pollination, biocontrol, insects serve as a food base for many animals and a couple plants, organic matter breakdown, soil aeration and nutrient movement within the soil, water filtration, propagule dispersal, such as seed dispersal, algal dispersal, fungal dispersal, medicines, shellac, dyes, jewelry, research models, and just their intrinsic beauty. And there are probably many more. And so when we talk about the potential of insects declining based on the massive numbers of species that they represent, but also representing all these ecosystem services, it's scary. So quite often insect decline is talked of being insidious, and the Webster Dictionary defines insidious as proceeding in a gradual, subtle way, maybe unnoticed, but with very harmful effects. So what's been some of the evidence? Well, one of the early studies was by Dunn et al. In 2010, and they looked at bird population census across Canada from 1970 to 2010. And what they did was they categorized birds into 8 groups. And when you look at this graph, on the x-axis is the years of the census, and on the y-axis is percent change since 1970. And the heavy black line is all birds. And what you can see is that raptors, waterfowl, colonial seabirds were all increasing in terms of the percent change. And forest birds, other water birds were pretty much staying the same. But what became apparent to them is that shorebirds, grassland birds, and aerial insectivores, these 3 groups depend on insects or other invertebrates. And so from that, their conjecture was that insects are declining, the food base of these birds, which then were leading these birds to decline. And so the points of their study really is that this was on almost a continental scale, that it was happening all across Canada, and there were implications that this was happening in the US as well. And it was verified across the whole northern globe. And their assumption, of course, is that the decline in birds that were insectivores was linked to an ongoing insect decline that people had not discovered yet. Well, the three biggies that caught people's attention were published in 2017 by Hallman et al. In Germany. And as far as I can tell, at least currently, that is the most cited paper of insect decline, with over 1,500 citations presently. Lister and Garcia in 2018 worked in Puerto Rico and demonstrated insect decline, and Sanchez Bio and Wickois in 2019 demonstrated insect decline in Europe and parts of North America. So we're just going to look at one of these, the Hallman et al. Study. And they conducted their study from 1989 to 2016, and they surveyed 63 nature reserves over this survey period. And that gave them 96 unique site-by-year events. And they used malaise traps to sample flying insect fauna, and they used biomass as a measure. And I'll get back to that a little bit later on. But they found a 76% decline from 1989 to 2016, 82% decline in summer flying insects. And this table shows for each year the number of locations that were sampled and the number of locations that were sampled previously at that year, and then the number of samples totally collected and their mean exposure time. In other words, how long they left their malaise traps out for days. And I want to bring your attention to the year 2014, because what this table is showing you is that locations were sampled, but usually they were only sampled one more time. So if a location was sampled in 1991, it was sampled one more time, maybe two times in this time series. But you can see that many of them were resampled in 2014. So this is their data showing time on the x-axis and daily biomass captured in malaise traps on the y-axis. And I've highlighted some of these pairs of samples from a particular location. And it shows that several were showing a decline. The yellow one is showing a stable population. This thick black line is the average. So it's the statistical model expectation of this data. And what they showed also in graph B in the upper right is that 93% of these sites, when they did a paired sample, had an intrinsic rate of increase that was negative, so that the populations were decreasing. So their points out of this paper was that they sampled many sites comprised of many landscapes, and their assumption was that sets of paired samples represented an insect community decline over a large region over time. And I'll revisit this, but also they use biomass. And biomass is a very advantageous measure to use if, especially, you don't have access to a diversity of taxonomists who can identify the diversity of insects that you're catching in malaise traps. Because biomass, all you have to do is collect the insects in a cup, dry them, weigh them, and you're done. But one of the issues here, and I have 1,000 to 6,000. So in Maine, I looked at Cecropia moth, and then I looked at Diptera that you're liable to catch in malaise traps in Maine. And the biomass of the Cecropia moss is 1,000 to 6,000 times the biomass of a typical dipteran that you'll catch in a malaise trap. And so you have to be very careful when you are sampling a wide range of taxa using biomass, because you could see that you might have started out with a couple of large moths. And if you lose those through your sampling, you might say that there's been 80% decline, but in fact, from an individual basis, maybe you only had, less than 1% decline. So you have to be very careful with biomass. Aerial sampling aphids is probably fine. So, and just the last study was a literature review by David Wagner in 2020. And he reviewed the conservation census data in the UK, which is a country that has the most studied insect fauna. And what he found on the graph on the right is showing 5 orders, and it's the percent of insect species are the red bars that are decreasing. And for the beetles, it's 60%. For the ants, about 45%. For the Lepidoptera, about 25%. The odonates, about 20%. And the Orthoptera, surprisingly enough, almost 100% are in decline, their abundances. Oh, this is percent of species. On the right, it's a percent of decrease in these orders over 40 years. And you can see again, over 40-year period, the beetles are about 40%, the ants 50%, the Lepidoptera 45%, and the odonates about 60%. And so, this suggests that there's been massive declines in some of the more common orders in the UK. But these declines are really difficult to assess. And there's been a call for conducting experiments because many of the alternative methods of assessing decline are difficult to navigate and maybe at best only guesses. But I've been thinking a lot about, well, what kind of experiments can you set up to look at community decline? It's a daunting task just to think about it. And I really couldn't think of any experiments other than a monitoring study. One of the common ways is to use historical surveys. The trouble is there's a lack of historical data, and there are several statistical issues, which I'll mention a few as we go on. And then there are current and future monitoring studies. Some of this is plagued by the lack of a baseline historical data, and so you're starting from scratch. Dry weight measures are often proposed, again, because of the lack of taxonomic expertise, unless the monitoring study is narrowed into one group of insects. And then there's a huge labor investment for monitoring because sample needs to be done across many sites and habitats, across several decades at least, and again, a lack of taxonomic expertise. So just conceptually, they're difficult to plan and can be very expensive. Well, some of the difficulties I'm going to talk about now, one is the form. So we have no knowledge really of decline dynamics for communities. We certainly know about declines of individual species and what form they may take, but for whole communities, we're not really sure whether they tend to be polytonic, fluctuating over time in decline, whether they tend to be more gradual, linear, or whether they tend to be more explosive in geometric or exponential, rapid declines. And the problem with not knowing this is that you can certainly do an analysis on dynamics that you have no knowledge of, but what happens is that you have to use less powerful statistical techniques, and then also your conclusions always have to be couched a little bit if you're not sure what the theoretical form should be. Sampling effort, unusual in many historical surveys to list how many people were actually involved in sampling, how many samples were taken. Proxy might be the number of towns or townships that are sampled. The insect survey by the Maine Forest Service recorded both sampling effort, the number of staff sampling, and the number of days that they sampled and the total number of samples that they collected. And in addition, there were records on the towns and townships surveyed, which is one of the most common pieces of sampling effort that historical databases include. And this is a relationship from the Maine Forest Service that's associating the number of staff sampling effort on the x-axis with the number of townships. And you can see, at least in their data, it was highly correlated. And so townships may be a good proxy for sampling effort. Of course, you can imagine a situation where only one town was sampled, and in one situation, only one person was sampling it, and another situation, you had 15 people sampling that town or township. And so it's certainly not always going to be the case where it's clear cut. But you can correct by sampling effort. And in this case, I'm going to show an example of beetle family richness. So this is a plot of the number of coleoptera families sampled from 1968 until 1998. And you can see that there's a clear decline in the number of coleoptera families. But when you correct it and adjust this data with the sampling effort of staff hours of sampling and the numbers of samples they took, you can see that the adjusted coleoptera families show no trend of decline. And this looked like what happened is that there was a lot of sampling effort in the beginning of the survey, but then when the survey was coming closer to its end, there was not as much staff sampling. And so you've got this distorted view of the richness. Sample time bias. This brings us back to the Hallman study in Germany. So this is a piece of data of mine over a 27-year period of sampling wild bees, broken down into 4 groups in a blueberry field in Winterport, Maine. So it was a single location over time. And let's say that we pick 1989 to start a sample, and we took the second sample in 1999. You have a drastic decrease in the abundance of the bees. But if you sampled in 2003, less of a decline. And if we decided to sample in 1990, and then for our paired sample sampled in 2005, we might assume that we actually have a population increase in this community. And so sometimes the time that you sample and take your samples, if you have only very few, can really bias the results that you obtain. And the last one I'm just going to talk about is knowledge of population dynamics. So this is the spruce budworm. And for a lot of individual species that are pests, we do have quite a bit of knowledge of their population dynamics. But for many species that make up our community richness, we have no clue of their biology or what their annual fluctuations might look like. And here you can see that the spruce budworm tends to go through huge fluctuations and almost outbreaks. And then for a while, it looks like there's nothing there. Well, if you have no knowledge of this, you might fit a robust regression. And what this does is it fits a line to the data by putting very little weight on these higher, more populous measures. And the conclusion here would be, yeah, despite those really high peaks, there's a decline. Well, you could also take a logarithm of this abundance data. Whoops. And if you took a logarithm, you might say, oh, yeah, this is definitely a decline of the log abundance of spruce budworm. But if you look at it a little more closely, you might say, well, gee, it's kind of odd that there's a bunch of abundance data points early on that are below the line. And then later in the survey, they're all above the line. Then even later in the survey, they're below the line. And at the very end of the survey, they're above the line. So something's kind of weird there. So if you have knowledge of the population dynamics of spruce budworm, you know it's an outbreak species, and it goes through outbreaks as shown here on this bottom right graph. And then there's a period of where it's in low densities. And now on this graph, it looks like actually the population's increasing. And we do know that we're entering another outbreak phase in the Maritimes and Maine. And so this is not in decline. This is just representative of an outbreak population. So without the knowledge, we can make some wrong conclusions. Okay, so now we're going to see about insects in Maine. And I have here the American burying beetle. It's critically endangered across a lot of northeastern North America. But the real question is the community in trouble? Not just individual species. I mean, we worry about individual species, but in Maine, we already know that there are species that we can no longer find. But with insect decline, we want to know, are whole communities, large taxa of insects disappearing? So the approach to exploring decline in Maine was a statistical analysis of historical surveys. And this study was due to a grant proposal submitted to the Maine Outdoor Heritage Fund by several investigators, and the project leader was Sarah Haggerty. And there were about 34 participants in this project to provide feedback, to help track down these historic databases. And there were several people that should be acknowledged that hunted down and found these historic databases. And Charlene Donahue was one. Ron Butler, who's listening on the talk now, was one. Philip Demarnier was another. And also the Leon Samides, I think, helped track down the Department of Environmental Management Aquatic Historical Survey. So with these surveys, the idea was to first do a community level analysis and to see if there was a decline in total taxa richness. So if it was a moth database, we were looking at whether there was a decline in the species richness of moths, but then also to look at the total abundance and whether there's been a reduction over a long time period of the total abundance of moths. But in addition, we wanted to look at multiple species or genera and to see if there was a decline across individual species and their abundances. And these usually were the common species. Spatially and temporally rare species are difficult, if not impossible, to evaluate decline. And that's because they may be in the environment, but they're so low density that they might be there, but you may never detect them. And yet, unfortunately, the rare species are the ones that are really generally considered the most vulnerable. And so those are the ones that sort of fall between the cracks quite often, unless there's a massive amount of survey energy put into them. So the data sets that I was given when I was brought on as a contract statistician to do this work were 18 in number. And out of that 18, there were 8 that really weren't possible to use. Either there weren't enough years in the survey, or the survey only included a few years, like the Proctor survey from the 1940s and 50s. And then that was it. That might make a good baseline survey database. Or some of them, the taxonomy was not updated, and I'm not of a taxonomist enough to do that. Or the survey was compiled by surveyors who did not collect the common insects. Maybe they wanted to collect an entire census of the species that occur in Maine. And so once they started finding some of the taxa, they weren't interested in those ones because they already had a record of them. And so they ignored them and collected only ones that they hadn't collected before. And so this type of database really doesn't lend itself to an analysis of decline. But we did have 10. And we had an ants database for Maine, compiled by Aaron Ellison. I had a lowbush blueberry insect database myself, covering between 30 and 50 years of data. A Maine Forest Service hoverfly database, the DEP aquatic macroinvertebrate database, a Maine Forest Service total arthropod database, a spider database, moths, the Maine Dragonfly Atlas, a Butterfly Atlas, and a little bit of the Bumblebee Atlas. And this is one of the very common spiders that occur in blueberry fields anyhow. One of my favorites, this thin-legged wolf spider that you come across quite often in blueberry barrens. So we're going to start off, we're going to cover 4 of the taxa out of those 10 that I looked at. And so the first one we're going to look at is the Maine Forest Service moth data set. And the survey years were from 1953 to 2020, 191 taxa were trapped over that time. But the database was, it surprised me that it wasn't well represented by a lot of the species. And what I mean by this is this graph characterizes a database. On the x-axis is the number of years that a given species was recorded in this almost 70-year time span. And you can see that most of the species were only recorded a couple of times in this whole 70 years. So the most frequent species occurrence only occurred in 2 years out of the 70. The median was 2.5, the mean was 9.2 years, and the maximum was spruce budworm with 56 years. So I wasn't able to do the community analysis per se on this, but instead I looked at the common taxa that had at least 14 years of collections, and there were 39 of them. And as it turned out, there was evidence of decline for almost 18% of that 39, 7 of them. Evidence of increase, 15.4%, so 6 of them. And so the conclusion, some species are declining in abundance, some species are increasing, about a similar number. So these are the 7 that were in decline. And first, it was the fruit tree roller, native species. The snout moths, a genus of snout moths. The spring hemlock looper. The hemlock looper. The spotted tussock moth. The large yellow underwing, which is interesting. It's a European native, so it's an exotic species here in Maine. What's interesting is that a species of concern in the UK. And it's becoming very, very rare to find it. And it appears to be dropping in abundance in Maine since it was introduced. And then the poplar sphinx moth. Well, let's look at some of the data from 2 of the species that I mentioned had declined. The first one is the hemlock looper. And you can see that over the years studied, it had one huge peak in 2000, and then pretty small numbers, very small numbers of abundance divided by trapping day, standardized by trapping day. But a robust regression shows that, yes, it looks like there was a reduction in their population numbers. If you looked at the logarithm of those numbers, it maybe becomes a little more clear that certainly it looks like there's a reduction. And really after about 2003, except for one blip, the numbers were very low. So that one does look like it's in decline. The poplar sphinx moth fluctuates a little more frequently than the hemlock looper. But again, the last large population increase was 1980. And then it doesn't look like it's really done much since then. And a robust regression suggests that, yes, indeed, it's in decline. And looking at the logarithm of those abundance data over that time period, certainly looks like it had been in decline from 1950s until 2000. So this is the type of data that was suggesting decline. Now, that's not to mean that if you look at the poplar sphinx and you see that last little data point, it's a little bit of an increase. Now, maybe the species has long boom and bust cycles and it will rebound in another 10 or 20 years. Maybe it's in high populations now. So you have to be careful with the assumptions of decline. So the ants of Maine, data from 1864 to 2011, 93 taxa. And for the entire data set, there was no decline in richness or abundance, but there were 4 species that were found up until 1998. And then after 1998, they weren't found. I also looked at some more recent survey data, which was part of the entire data set. And I'll show you why I looked at that in a minute. But richness declined, but not abundance. And there were 4 species abundances that declined, and 1 species where the abundance increased. So this graph on the left is just the total abundance divided by township of the ant survey from 1864 to 2011. And that same data from 1940 to 2010 is the species richness per township. And it looks like there's no trend there, but if you look at the data from 1998, it certainly looks like there might be a decrease in ant richness over that last decade. And so this shows on the bottom, just that, isolating that group of data points, the species richness per township. And it certainly appears that there's been a decline in ant species richness up until 2012. So I mentioned that there were 4 species that were not found after 1998. If you look at the distribution, they're fairly widely distributed out to the Midwest or down to the South or over most of the US, and not in really unusual habitats. Blueberry barrens, prairie, sort of pine woodlands, mixed forest, sandy soils, or richer organic soils. But one thing is that their life histories seem to be somewhat either dependent on another species or they tend to be very small colonies. So Formica dolosa is a host to slave maker ants. Formica nepticula is likely a parasite of an unknown Formica. The host isn't known. Myrmica neotica, it's not well known, but it lives in very, very small colonies of less than 20 individuals. So could be more likely to exportation. And then the winter ant, or the false honey ant, Prenolepsis impairs, it's maybe its life history that has fooled us into thinking that it's declined. It lives in very deep nests, greater than 60 centimeters deep, and it forages only during the fall and the spring. And when it does forage, it's either just crepuscular or nocturnal. And in the summer, it stays underground in its nest, it seals it off, and it has a replete stage, which is a morphological stage that is the bottom picture. And the abdomen is filled with lipids, and they hang from the ceiling of the nest. And during the summer, instead of foraging, they feed on the lipids from the abdomens of these repletes. So it certainly wouldn't surprise me that we still have it, but just people haven't been out sampling ants in the late fall or early spring at night. I mentioned that there were 4 species that declined in that group of data points from 1998 to 2011. And all 4 that declined are very small colonies of less than 50 ants in a colony. And for instance, Myrmica punctiventris actually, their colonies live inside acorns. So their nest area is quite small. And then the species that increased is the odorous house ant, a pestiferous exotic ant. I have two of these species that I have asterisks on, and I have them listed here in decline, but these regressions were affected by a single data point, and this is called being leveraged. And that data point is probably a real data point, but it might just reflect an tremendous increase in abundance that a tremendous decrease in abundance that resulted in a regression that suggested that species was in decline. Okay, the third group were the aquatic macroinvertebrates, and the data went from 1974 to 2019. The identifications went to genus for most of the taxa, and there were 826 taxa. And for the entire sample period, using all sampling methods and all stream classes, we found no decline in richness or total abundance. And looking at individual taxa, again, utilizing all sample methods by accounting for the different sample methods with a mixed model regression, it looked like about 3.6% of the abundance, 3.6% of species, their abundance had declined, and 8.1% of species, their abundance had increased over time. And here's one example that decreased. It's the genus Glossoma, which are the saddle casemakers or the little black caddisflies. And you can see that there's certainly a lot of scatter, but in general, it looks like it's a declining, the average abundance for each year looks like it is declining over time. But the conclusions appear to be dependent on the sampling device used. And so I looked at two very similar passive collecting devices, are rock baskets and rock bags, which are put into a riffle area in a stream and are usually collect macroinvertebrates for about 3 weeks, and they're extracted from the stream and then combed of all the macroinvertebrates. And for the rock baskets, we found an increase in abundance and increase in richness over the time frame, and similar numbers increased in abundance of taxa and similar decrease, 4.8% increased, 3.5% decreased. For the rock bags, there was no decline in abundance, but a decrease in richness. So even though these are very similar types of sampling devices, it looks like the sampling device used will alter your conclusions. And 2.9% of the taxa increase in abundance and 3.7% declined in abundance. So overall, as many taxa appear to be increasing in abundance as decreasing in abundance in both of these scenarios. And the last is the Maine butterfly survey, which Ron Butler has been in charge of and highly influential in putting it together. And it included data from historical surveys as far back as 1864 up until 2020, included 121 species, 91 of which had subspecies identified. 20% of the species, approximately, are of concern or threatened. And overall, species richness and abundance corrected by township, we find that species richness declined since 1864, but there didn't seem to be any decline in adult abundance. And this graph looks at log richness per town. And then the graph on the right is log abundance per town. And you can see that log abundance is pretty flat, but richness is decreasing. So while the number of species is declining, the abundance per species that remains is increasing. Species of concern, there were 10 out of 19 species that showed evidence of decline. And these are the 19 species of concern. And any of these species that either have a 3 or a 4 have either are highly leveraged on the regressions. As I mentioned, they're influenced by just 1 or 2 data points, or they had very few data points over the whole time series to really be confident in their conclusion. But let's look at an example of 2 of these species that were shown to be in decline. One is the crowberry blue, and this is the abundance per town from 1985 to 2010. And then Clayton's copper, which I've studied a bit in northern Maine in some of the bog areas. And this is the abundance of Lycaena dorcas per town. And you can see that it certainly looks like it's been in decline. So some of these that aren't leveraged or have enough data points to look at certainly do look like there's been a reduction in their abundance. 18 of the 32 most abundant butterflies, and I classified abundant as having at least 1% relative abundance, so a percentage of the abundance of the whole, showed evidence of decline in abundance. And here's an example of the Atlantis fritillary. And on the left is just the population fluctuations over time. And then on the right is the logarithm of its population abundance corrected by town. And you can see that it certainly looks like the abundance, the log abundance, is declining. More recent surveys during the Maine Butterfly Atlas 2007 to 2020 showed that species richness actually marginally increased, which means like a p-value of a little greater than 0.05, but less than 0.1. But total abundance looks like it might have declined. And here's on the left, the species richness divided by towns as an adjustment of sampling effort. And then abundance divided by towns certainly looks like it has declined. Looking at the commonly abundant species, 9 of 28 showed potential decline, although only 2 of these 9 that showed potential decline had significant p-values of less than 0.05, meaning that they had strong decline relationships instead of marginal. And one of them, strangely enough, is the imported cabbage worm, an exotic imported from Europe, which is a pest species, but it looks like it's at least since 2016 or 2015, maybe going through some decline. The northern spring azure, again, looks like it's had 4 years from 2016 to 2020 in decline. So these more recent surveys, the nice thing is that I was able to do an analysis by town. And when I looked at by town, it suggests that butterfly declines often occur locally, meaning that you might have a neighboring town where a certain species of butterfly is stable, and in another locality not far away, it's in decline. So aggregating species and occurrences and abundance statewide actually might do more harm than good. It might mask declines and extirpations that are occurring locally. And so that's something that maybe should be looked at more carefully, because all of my analyses were aggregating over the towns. So in summary, we looked at the moths, the butterflies, the dragonflies, other flies, native bees, bumblebees, just in a cursory manner, ants, spiders, and the aquatics. And the second column lists the number of taxa, the survey period, and then I have, under the richness and abundance, a red D if it was found to be in decline. An orange D for the native bee abundance was because it was sort of just marginal. And then the blue eyes suggest increases. And so I did an aggregation, which really doesn't mean much, but it's more of a summary. I aggregated sort of these richnesses and suggests that 75% of the analyses of the taxa that were done had the red Ds, and only 12.5%, or 1 out of the 8, had increases. On the abundance, 62.5% showed a community-wide decline, and 25% showed increases. Looking at abundance of species of special concern in the butterflies, about 42.9% looked like they were in decline, and 13.3% of the odonates were in decline. And then looking at the common species, the last column, you can see it ranged from 56.3% in the butterflies down to 3.6, 3.5, 3.7 in the macroinvertebrates in the aquatics. And this actually has been somewhat of a trend globally. Those people who have looked at macroinvertebrates, aquatic species, have found that the decline has been less occurring. Not that it hasn't occurred, but it just hasn't seemed to have been as drastic as the terrestrial. And I do have starred for the spiders that the only declines I found were in spiders in spruce understory habitat, and not in blueberry barrens, not in any of the old fields, but just in the spruce understory habitat for some reason. So I guess in closing, it certainly looks like we have concern. These analyses are far from exact, but they do certainly suggest that we're having issues in Maine across a lot of our taxa. So the last part, I just want to briefly touch our potential causes. And Wagner et al. In 2021 published a paper titled Insect Decline in the Anthropocene, Death by 1,000 Cuts. And so their main point in this paper was that insect communities are experiencing multiple stressors simultaneously, and it's probably folly to look just for a single stressor. So urbanization, light pollution, these are many of the ones I've listed here that have been brought up mostly as hypotheses. It's very difficult to assign cause and effect. It's to decline. It's difficult enough even just to assert decline. But vegetational landscape succession, this has happened in Maine since the 1800s, where Maine had huge cleared areas, and now 93% of the land cover in Maine is forested. Land use changes and development, little bit in Maine. Fragmentation, pollutants, probably not as much in Maine, but in some areas, some of our streams, certainly some urban areas. Invasive species for sure. About 25 to 30% of our plant species along roadsides are invasive or exotic species. Climate change for sure. Light pollution, maybe in some of the more urban areas. Highway mortality, electromagnetic radiation. There's a paper that just came out recently about this, and it's been poo-pooed for the last couple of decades, but people are considering it a little more now. Sanchez, Bio, and Wickers, they surveyed the literature, and what they found for the main factors that are associated with insect declines in their studies were intensive agriculture, number one, 23.9%, pesticides, ecological traits, so mismatch of ecological traits to a changing environment, urbanization, fertilizers, deforestation, wetlands, and then after global warming, the percentage of importance or association with insect decline becomes less and less. Well, I guess I'm going to close it there, but I'm just going to finish by saying, I guess I was really surprised at these results because I have always thought of insects having short generation times, high fecundities, of being able to undergo rapid evolution. And I was really surprised that apparently there are many species that aren't able to adapt to the stressors that are making them decline. And part of that might be that there's multiple stressors, and it's a lot harder to adapt to multiple stressors instead of just a single selection pressure on you. So I suppose with that, I'll open it up to questions.
So if you could put your questions in the chat, or there's too many people for me to see if you've got your hand raised, but you could try that under reactions. And maybe you'll come to the top of the screen and I can, or just speak up. That's also possible. So Frank, I've got a question that this seems to be a super hard thing to actually get your hand on because in any ecological system, there's so many interactions happening that it's hard to predict the cause of a change from year to year of any particular species. Do you feel that the research is robust enough to overcome this?
For finding causes, you mean?
Yes, yes.
Boy, it's so one paper I read on the causes, they looked at Lepidoptera abundance in the northern part of the UK and the southern part. And the northern part, there was no decline. The southern part, there was a decline. And they looked at light pollution, and it was much greater density of lights, and the lumens were greater in the southern part. So they associated that, and they said it's due to light pollution. I think many of the studies are like that. Some use statistical methods to partition out, if they also have that data, partition out what percentage of the variation in decline is due to these factors. But, you know, even that, the trouble is those factors often are autocorrelated with one another. They're so they're not singly explaining the variation. And so even using very sophisticated statistical methods, I think we're only coming up with an approximation. But one of the calls for doing experiments was to basically try to focus on causes and not so much the declines, but focus on causes and see if one could do like a five-year study on there's been some good studies done on highway mortality, where people have actually put sort of nets and screens on the front of vehicles and gone up and down, you know, freeways and rural roads and correlated the mortality of insects that they're capturing on the screens on the front of their vehicles with traffic volumes. And so maybe there's some of that can be done. And but my feeling is sort of what you're suggesting is that it's hard enough to do for a single species, to do sort of a life table analysis and look what's causing decreases in abundance. But to do it for a whole community, especially when the taxa are in different orders, is mind-boggling, really.
Sure. All right. Well, thank you very much. There's a couple of comments saying thank you and excellent presentation. I certainly can echo that. But anybody else have any questions?
Yeah, I do. Just a
Oh, there you are.
yeah, in the dark here. Yeah,
You're in the dark.
Thank you, Frank. That was really great. That really does give quite a few stimulates a few questions for me anyway. One of them, at least, was that, and I know the data are pretty limiting, and you can only conclude what you conclude from the little bit that we have. But at least one of the species that you showed, you thought that there was some evidence that I think it was a moth species or a butterfly species is showing more rapid decline since 2016. And I'm wondering if that's something you think could be parsed out from the data that we have. Sorry, my face is still in the dark here, but there we go. And if so, is there reason to think that maybe we are going to start seeing more rapid decline, or is it just that it's just too hard to parse that out from the data?
No, I think you can parse it out, but I think you're left with the variables that you select to measure. So for instance, one thing that comes to me from like 2016 until about last year, actually, we had a series of continuously dry summers. I mean, like really dry, especially in like 2019, 2020, 21, were really dry. So much that a lot of the plants stopped secreting nectar. And so that comes to mind. So yes, you could look at that, but associated with that, they were also a lot hotter than normal. So is it the drought or is it heat? But I think it's worthwhile to maybe do that, is to see what data exists. The problem is, for a lot of those, they might have been from different towns. And so you'd have to, it might be that you wouldn't quite have enough data because, let's say 4 or 5 of those points, they might have each been from a different town, a different year. But so it gets a little more, you're on a little thin ice that way. But yeah, I think that it would be really good to try to get at least, I think you can get abiotic weather data. It becomes a little harder to get, whether animals are being exposed to toxins, maybe a little harder to get levels of light pollution, habitat changes, land use changes. I suppose people with really good GIS skills could milk some of that out, as what's been as long as we have updated layers to look at the habitat layer changes over time. I mean, sort of what Roger asked, there's maybe a lot of confounding factors, but it's worth a try for sure. But I don't know if when you do it, if you just say, well, there's a tendency there, but I don't know. But you won't know until you do it. I think Ron Butler wants to do it, and I would encourage him to do it.
I see that Marnie has
Thanks, Frank. I appreciate
Marnie
that.
has her hand raised. Marnie, if you want to unmute yourself, you just did. Okay, go ahead.
Yeah, we have the last 10 years, moth photographs, several thousands of them, from 3 black lights sheets that are on the edge of the Rhode Island Nature Preserve. And we're in the process of putting this data as a collection project on iNaturalist. Do you have some other suggestions as to what we ought to do with this data so that people in the future could have the benefit of it?
Well, so you were going to have the photos identified, is that right?
Yeah. Yes.
And they're moths. So were they captured in light traps or?
No, they're photographs.
Oh, photographs.
Of the sheet, yes. And iNaturalist makes suggestions based on the photographs that it looks like thus and so. And if we agreed that we thought that is what we had, we'd put that down as the species or the genus or however close we can come.
Well, I'm probably not the one to ask. I bet there are other people here that might have more knowledge about that. But let me just ask you, did you take a picture of everything on a sheet, or was it just random things on a sheet, or was it likely that some of the more attractive, stunning moths were taken more likely than sort of a microlep or something on a sheet?
No,
or...
we did microleps as well, and we tried to do a picture of every one. And by the way, our Gmail address is Marnie, M-A-R-N-I-E, Crowell, C-R-O-W-E-L-L @gmail.com. So if any of your watchers want to tell me their brainstorm of what they think that we should do with this data, that's our winter project, is putting this on iNaturalist, because that's all we can think of. We could send discs to people, or I don't know.
I mean,
it's more too many thousands for, we're 84 and 90. It's too much data for us to deal in many ways. My husband is a retired ecologist, but we've been living next to this preserve for years. So we've been aware of what changes there have and have not been, which is right here. There's hardly that.
On paper for common species.
And
Marnie,
yes.
This is Roger. I can make a suggestion. I think you're doing exactly probably the best thing is to put every one of those observations on iNaturalist. It's a really powerful tool to look at changes over time. Now, how you can actually document what you did, I'm not sure the best way of doing that. It really, it's worth documenting exactly how you collected some of the questions that Frank asked, and perhaps posting that in one of the chats on iNaturalist, and even asking, okay, what should I do with this?
That's a good idea. Yes, I do have a picture of one of our lights in the sheets, and it's because we've done the same thing for each of the 10 years and all of the seasons, basically from when the maple sap starts running till into November. We're still doing it this week. We've been still getting a few moths. So it's a lot of data.
You have tens of thousands of photographs?
How many thousands would you say we have, Ken?
Well, yes.
He says tens of thousands, yes.
Yeah. And so I'm not as familiar with iNaturalist. How long, I mean, some of them might be identified, but some might not.
Some of them are just until other people look at the photograph and say, oh no, that's not a thus and so, it's somebody else. And when they get 3 people agreeing, they call that I don't know, special category of research data.
Yeah.
So.
Yeah. Oh, it's great. It almost would be wonderful if there was someone out there who could build an AI algorithm that would classify them. You know, they certainly have those for plants, and they're fairly accurate, and it would be nice that they could automate it and do 10,000 in a day or so, and maybe there's someone out there who would do that, but I don't know, because it may take a while for, you know, if you want to complete. See, I think what might happen, maybe I'm wrong though, but on iNaturalist is that a lot of the common ones would be identified first, and it would take longer for some of the other ones, and maybe some of the more spatially rare ones, and yet those might be of particular interest. So it may just take a while before the data is ready, but once it is, I think you'd have a fantastic data set to look at.
And Catherine somebody just asked for what our username was. Would you please put that back on the screen so I can get how to answer that question for you? What's your iNaturalist username?
You used it.
I haven't used it recently.
Ron, is there something you wanted to say?
I just was going to add that iNaturalist already operates with an AI. So if you put a photo up there, the AI is going to make its best guess at what it is based on the increasing by the minute, millions and millions of photographs that have already been posted
Right.
and vetted by experts. And so the AI was,
So fast.
iffy at first, but it's getting better and better.
Oh, good.
That said, there are some things that won't be identifiable based on the need to do microdissections or have a variety of different macro shots of the beast. But iNaturalist seems to be now the go-to site for not just insects, but for you name the organism. I mean, it's been an exponential growth since it went online in 2011.
Yes.
I mean, I
great.
routinely visited each year to harvest odonate and lep and bumblebee records. So sometimes you can identify them no problem from the picture. Other times you can't because it's a crappy picture. So it's just life.
We have a few crappy ones.
I can also say that it has gotten so much better. I can go back and take ones I couldn't identify 2 or 3 years ago, and then repost them, and now can identify them through the database in iNaturalist. It's kind of like the Facebook of
Yes.
nature photography right now. It's just taken over from everything else.
Thank you, everybody.
All right, Frank, thanks a million. This was just tremendous, and thanks to the audience for your questions and comments. Let me just look and see. So Marnie, there's somebody in the chat who's willing to take a peek at your iNaturalist pics as well. So you might take a look there.
Okay.
All right, with that, I'll say goodbye to everybody, and thanks again. And a reminder that on I think the 9th of January, Charlie Eisman's going to be talking, and his will be really worth hearing, and you all get announcements about that.
Thank you very much.
Thanks
Thank
for
you. Bye.
everybody.
Bye-bye.
Have a safe walk back to your house, Frank, in the dark.